I tell a bit of what we are doing here in Sweden. So the neighbors of Passy, hello, to close by. And then, yeah, what kind of projects we are having, how we are working, and then happy to take some questions also. So indeed, we are an entire team looking at bioimage analysis, working in bioimage analysis support, and mainly located here in Uppsala in Sweden. And then researchers are so we are a national facility, meaning researchers are from all over Sweden. And then they contact us, we help with some smaller consultations or set up entire projects where we try to use kind of state-of-the-art analysis tools of the community, and also build our own tools and give workshops and all these things. We are connected to research groups, so there's Carolina Weber's group here in Uppsala, and then by now 2 research groups. Giselle Miranda, who was a member of the facility and has now her own group, and then Kevin Smith, who is also a professor at KTH in Stockholm. And so how does it look like? So we get a lot of different kind of support projects requests, because researchers all over Sweden, so they have very different research areas and research questions and modalities. That's very cool, of course, for us. And of course, there are some trends, and I guess some things are even already kind of routine by now. So like AI and deep learning is a thing, and I saw a bit like the environmental or satellite images right now from Passy, and I guess we also get a bit more of these photographic images by now. That's not fluorescence microscopy, where people want to segment something. So a bit more environmental science arriving us. And then we, of course, deal with 3D huge data sets like organoids. So this is, I think this I'm showing quite often, but we have similar also screens for this, like how to deal with these kind of 3D light sheet mainly data. We do have a focus on spatial omics, spatial biology, and then also all kind of different data can arrive to us. And then, so it's a lot of image registration, not only on these more foreign image modalities, but also, of course, on like spatial omics data. We also look a lot in how to combine different modalities there. And I looked a bit on our KPIs, the key performance indicators from last year. And so we were helping 44 researchers, like either by a short consultation or like a longer project, long projects. And then we were involved in like several workshops. Mainly it's like in microscopy courses, and then we give the image analysis part. We do have a YouTube channel. I have some links in the end. And then I will talk about tissue maps, or you will see also in the conference more about tissue maps. So that's interactive viewer that we are co-developing, and this one has reached quite nicely amount of views, so I want to say of downloads per month. And then I think we try to really be involved in the community and general pipeline development that is around. And I already said that we are quite focused on spatial biology, spatial omics. And I guess that came also because the experimental part has been pioneered a lot in Sweden. So there's a lot of happening in Sweden, and super exciting. And then also like the tissue maps part is really like development is part of this kind of area. And like in the microscopy audience, it's nice to explain this kind of background of spatial omics. So the idea is basically that you want to have you want to image your mainly tissue images. This is usually with a lot of channels, basically. That's how I used to explain it. So you want to show many genes or many proteins, and then you reach like the spectrum by like the number of channels, number of fluorophores that you can use, and also because of the spectral overlap. So you try to like different methods of overcoming this, like mainly like cyclic imaging of the data. And then you can end up with like 100 channels of different proteins or different genes in like one tissue section. And then one of these methods is what's called in situ sequencing. I think I never liked so much the name, maybe I shouldn't say that so loud, but I think it's mainly about, here it's about like visualizing RNAs in the tissue, so very cool. And what you sequence here is a small barcode. And so you have a barcode, and then this one you can kind of encode with like 4 fluorophores, and then you can like visualize again like hundreds of genes in one tissue. So it's very cool. And then here in Sweden, there's Mats Nilsson, who was really pioneering this. And then this is like then also delivered as a service on the experimental part in SciLifeLab. And then Carolina was also very early on involved in the analysis of this and the visualization also of this data. And so Carolina, in her lab, like started the tissue maps project, and then now as a facility we are involved in the development. And you will hear about this more by Christoph. I have the date in the next slide. But what it is really like an interactive viewer, a GPU accelerated in the browser. So you can load the images and some markers, and you can really millions of markers. You can zoom in, out, and inspect different regions. And export also the gene compositions of different regions and so on. So the aim mainly is to really allow if we have multidisciplinary teams with pathologists really being the expert of the data, that they can easily handle the data, and then the bioinformaticians can do more downstream analysis of that and give it back to the researchers and so on. And this is how it looks like. So here we are zooming into a part of that and switching on all these genes. So millions, you can see here, like 2.4 million markers. And then inspect like different single genes of how they're expressed. And there will be a presentation on Friday in the tool exchange session at 2:20 by Christoph. So he's the main developer now. And then you can hear more about this and hopefully try it out. Looking a bit on the time, so we have done also a different method, is the spatial proteomics. So again, we are wanting to create like really multi-channel images, tumor regions, but this time on protein level, not so much on RNA level. And then again, there are these cyclic methods that we always look at only maybe 4 markers and then wash them and then look again for new markers. And again, this is a method provided as an experimental support in SciLifeLab. So we get high quality data here with Charlotte Stadler heading this unit. And then what we did here is trying to give some resources to researchers that we saw where we saw like reoccurrent problems. So we have a YouTube video, how to deal with this data in QPath. And then we also have like course material on GitHub that you can follow with a bit more advanced analysis pipeline. So really using like pixel classification or object classification and strategies of how to build those. So these you can find like either on GitHub or on the YouTube channel. And then as a facility, we really try to be have the overview of what's around and try to find the best tool. And then we also can know about and use it, MC Micro, which is a next flow pipeline for this data. We have there's Steinbock, which was the main developer, Jonas Windhage, which is in our unit now. And then there's also PyPix, which is a tool developed in Emma Lindberg's groups, also a neighboring group. So what we try to do then here is kind of provide support really on this. Like if people want to use this, we can help them with input. And then for the PyPix part, what we did was that the PyPix output can be easily loaded into tissue maps. And then so people have an entire workflow then. So what you can see here is such a multiplex data set. So you can see here now this mini viewers of the different channels, a bit like in QPath. So you can see all the channels in parallel and then zoom out and zoom in. And then usually what you do with this data is do some clustering of the cells by their features. And then this is what you can see here. And then you can also look at UMAP space of the features and then interact with all these different channels of information, let's say. And then what you see here is, for example, interaction. So what we do, I mean, of course, looking at the spatial part, the main thing is that you want to see it like neighborhoods and which cluster of cells interact with which other cluster of cells. And this is also possible here. So, and then for this, we also gave a workshop last year at the I2K Images to Knowledge Conference. And then also all this material is online. So if you like to learn more, if you think this is useful, then please have a look on the GitHub, and then you can hopefully find it nice and then use it by yourself. And then another spatial method is, of course, spatial transcriptomics. So here, what is the kind of multiplexicity, how to say, is achieved by classical sequencing. So what's happening is that you have these spots, and each spot catches RNA, and then it's kind of given to a classic next generation sequencing machine. And then basically the spot is also again, so that you know which RNA was detected in which spot by a barcode. Again, a method highly pioneered here in Sweden, and then again also provided as support by many. SciLifeLab, and Anja Metzger is here the head. And then what we did here is teamed up with others in the NBIS, the National Bioinformatics Structure in Sweden, to build a NF core pipeline to really like have a pipeline for this kind of Visium, that's machine data, and do all the quality control steps, and then also have a file that you can easily load in tissue maps and inspect, and then have like a really nice, clear pipeline to use. And then, so that's the cleaning lady. So that was about the spatial omics part. And then I also wanted to mention that indeed we also get like spatial information by now that is kind of a bit different. And this is kind of also going into the satellite images and environmental data. And there we have a project starting up with a SciLifeLab PI, Tobias Andermann. So what he wants to do is to look at, like, to combine really very different kind of information. So kind of samples that they take on site of kind of which species are, or like which environmental factors are given, and then combine it with satellite images. So there comes then the image analysis into play, and then combine all this using CNNs, and then get a better description, also predictive power of how it looks like and this is a project then with Swedish data. I'm also wanting to shout out to upcoming activities. So there are two things. Like one is the Advanced Methods in Bioimage Analysis EMBO practical course, which will take place in September. I'm super much looking forward to that one. You will learn a lot about really kind of the cutting edge questions that are around, so also spatial biology, or you're looking probably at spatial data and segmentation and all of these nice things. So application portal is already open, and there will be a selection process, of course. And then I also wanted to shout out to the Globia's BioImage Analysis Conference taking place in Japan in October, which is like a training school, hackathon, tagathon, symposium, and so on. So also a lot of interesting resources here. And then that's already, I kind of targeted to 50 minutes to have some space for questions. So if you want to get in contact with us, then you can find more information on our website. And then there you can also really find links to the YouTube channel and to our project website if you want to scroll a bit into what we're doing and how support works for us. And then that's already it. So thanks a lot, of course, to our funders and then to a great team. And yeah, it's fun to work on these problems in a team together. So it's really nice.